All right, thank you for coming. And it's a pleasure to introduce Andrew Fung to give us a seminar today. Andrew is a senior associate consultant AI. I'm not quite sure what that is, but I'm new to Mayo, so I get a free pass. He's a member of the Department of Radiation Oncology. His education was done largely at the University of Cambridge. He received a bachelor's and master's degree in 2018 in engineering and computer engineering. He stayed at Cambridge, good place. He received his PhD in 2022 in machine learning. And then prior to joining Mayo, right after graduation, Andrew joined Microsoft as a senior researcher right in 2022. And I think he worked at my conversation with him, he worked on a number of different problems. And one of the things that probably was most, at least impactful to a biochemistry and molecular biology audience is a really important paper that just came out this summer in Science. Basically, it's a deep learning method to try to make predictions on relative free energies of protein ensembles. So for those of you who are not aware, proteins are more than static snapshots. They have to jiggle and wiggle around, as Richard Feynman said, and that's important for their functions. And we really are interested, both from experimentalists as well as computational chemists, understanding those free energy differences, because those are important for understanding things like enzymatic cycles and so forth. And Andrew's paper, which he was a first author on that paper, has been downloaded a number of times. And their error rate for predicting free energy differences is 1 kilocal per mole, which is really impressive. So that's less than an order of magnitude. So it far passes any other predictive things that have been done prior to that. That paper has been downloaded 30,000 times since July 2025 when it's been released. So it's been a really impactful paper, adding to this kind of revolution in deep learning methods that in biochemistry, at least, that really probably started with AlphaFold a few years back. So Andrew's joined Mayo in January 2025. So earlier this year, he's my senior at Mayo. And he's been working on several different problems in AI. Hopefully he'll tell us a little bit about that. One thing I noticed from his CV, and I watched a lecture or two, which is that he created a faculty development series called Understanding AI from Scratch. And so I think it's 6 or 7 lecture series, and it's really great. And he's a really outstanding teacher. And I think you should try to check that out if you're interested in understanding a bit more of the nuts and bolts of the black box of AI and machine learning and deep learning specifically. So with that, Andrew, thank you for coming here, and we'll look forward to your seminar.
Thank you so much for that wonderful introduction. Here presenting. I've not talked about this work for a while because most of my work in radiation oncology hasn't really touched on structural biology, but it's nice to be back in this world a little bit. A lot of this was covered by Nate, but yeah, I'm a senior associate consultant in radiation oncology. The work I'm going to describe here was all done in Microsoft Research, so that's really where this is coming from. My background is in machine learning. I spent some time in Google DeepMind as well. All this to say that my background is really in deep learning, and I'm kind of I tip my toes a bit in biochemistry, so don't expect me to know the ins and outs of that, but I can explain the deep learning architectures. If you want more information about what I do, you can look at my website. I'll share these slides later if you want to look at any of these links. Like Nate said, this talk is going to be about a model, a deep learning model. In particular, this model has a cute name. It's called BioEmu. It's short for a biomolecular emulator. The whole idea here is that we're not going to be predicting. We don't want to make the claim that we're predicting everything perfectly accurately, but there's a niche for emulation, predictions that are good enough that can happen really, really quickly, and that isn't really currently filled by many existing methods. So BioEmu, in a nutshell, it's a deep learning model that takes protein structures as an input and outputs a distribution over 3D protein structures. So I'll unpack this diagram a lot more, but the high-level thing to keep in your head: you give BioEmu a protein sequence, it goes through some deep learning stuff, which I'll talk about, and then once it's done processing, you can sample from it computationally, pretty cheaply on a single GPU. You can get thousands and thousands of structures. And these aren't all going to be the same structure. They're all going to have little variations, conformational variabilities. And from these structures, you then interrogate properties that you care about. So you might care about the folding free energy. Well, folding free energy, if you know the distribution of structures, you can compute that. You might care about the different conformations a protein might take. There might be cryptic pockets. There might be domain motions you're interested in. There might be mechanisms for observed experimental, effects that you want to hypothesize. That's really what BioEmu is trying to solve. So yeah, and these distributions, they're trained to really emulate this physical conformational variability that exists in a natural world. It predicts multiple conformations, not one, like AlphaFold. And we really hope to capture something of the thermodynamics of what's going on. So not just here are 5 conformations, but some kind of estimate of the relative probabilities of each conformation.
So it sounds like it's trying to imitate a molecular dynamics simulation.
Exactly. So this work was done supervised by Frank Noah. He's really done his entire career in molecular dynamics. And what we really aimed to do was say, let's do molecular dynamics faster and in a generalizable way with deep learning. Let's do for molecular dynamics what AlphaFold did for protein structure prediction. Now, many caveats on that, and I'm very happy to dig into the issues with that later. But yeah, like Nate said, this is a paper that was published recently in Science. So let me talk about, give a framework for today's talk. By the way, please ask questions at any point. If you have a question, probably other people have questions too. And yeah, stop me at any moment.
Don't worry, we will.
That's how I like talks. So first of all, some background, AlphaFold and the deep learning revolution, in case you haven't heard, deep learning exists. The next thing, I'm going to talk a little bit about AlphaFold and how it works. Just a quick survey. How many people here have heard of AlphaFold? How many people have looked at an AlphaFold paper in Nature? How many people have looked at the supplementary material of the AlphaFold paper in Nature? Okay, great. So that gives me some sense of the audience. That's great. I think that's what I wanted. I'll talk a little bit about how structure is not enough. Probably you would know more about this than I do, myself coming from a deep learning background, not a computational biology background. Then I'll finally get to talking about what BioEmu does. I'll talk about the training data we accumulated for it. I'll talk about how we're using a diffusion model, kind of what that is. Has anyone who has heard of diffusion models in machine learning? Okay, good. About half the audience. Then I'll talk about some capabilities, what BioEmu can do that's actually somewhat useful. And then we'll see if we'll have time for Q&A at the end. Let's start with AlphaFold. I don't need to tell you that protein function, at least a lot of it, is mediated through the structure that a protein takes. And a protein structure is more or less largely determined by its amino acid sequence. And the problem that AlphaFold and much of structural biology tried to solve was, given an amino acid sequence, what is the 3-dimensional structure of the protein that it folds into? So you have some kind of sequence. This is just some sequence I grabbed. And then you have all these traditional, reliable to varying degrees ways of obtaining the structure. So you can do X-ray crystallography, which is extraordinarily time-consuming. You can have an entire PhD to crystallize one very hard to crystallize protein. There's cryo-electron microscopy, which I take it is something that people here are interested in. NMR spectroscopy, honestly, I don't know much about these methods, except that I know that they take time. And then at the end of it, you get a protein structure out, and you can do some useful structural biology interrogation with it. Now, I don't need to remind you that Google DeepMind famously cast the protein structure prediction problem as a supervised learning problem. So supervised learning means you have some input, you show it to a machine learning model, you have some output, you have lots of labeled examples. The model learns the association between input and output, and then you now try it and then you input. So where does it get all this data from? Well, it came from the wonderful protein data bank. For decades, researchers uploaded their protein structures obtained at great cost over the course of many thousands of PhDs into the protein data bank, and anyone could download them for free. It was an extraordinary, what John Jumper called as a data overhang. Imagine a cliff and you're kind of overhanging the cliff. There's just all this data that was all in the protein data bank overhanging the cliff, and what AlphaFold did was it took advantage of that wonderful data overhang. And the key breakthrough that made this data more than the sum of its parts was deep learning. AlphaFold really through and through was a deep learning innovation. And what it really tries to position itself as is, instead of doing this really expensive experimental techniques to obtain protein structure, if you solve the supervised learning problem, which AlphaFold 2 arguably did to a very high degree, then you can do this much more quickly. So I'm going to go through deep learning very quickly in a nutshell, just to get us on the same page. But I'm sure you've heard much of this before. deep learning is a synonymous term for the use of artificial neural networks. You can draw them in many ways, but often you'll have a diagram like this. What they really are is they're mathematical functions with millions of numbers that define their behavior. These numbers we call parameters or weights. And at a high level, these numbers determine how the neural network behaves. You adjust all of these a little bit. You start by choosing the parameters randomly. And of course, if the parameters are garbage and random, then the outputs are garbage and random. But you then have an optimizer whose job it is to automatically adjust the parameters to fit the example data you show it. In this case, the example data was the protein data bank data, the structures there. So this optimizer automatically adjusted the millions and millions of parameters in AlphaFold 2 to fit protein structure prediction data. Then at the very end of it, once you're done doing that, you apply the function to brand new data. So a brand new sequence, AlphaFold 2 was never saw during training. And in the case of AlphaFold 2, what they did was they took advantage of the CASP competition, which is a protein structure prediction competition. Every year people deposit structures into this competition that they don't publish in the PDB. And then AlphaFold 2 was given the sequences, but not the structures. And it predicted these structures to a very high degree of accuracy. And the organizers of the competition considered the competition's problem solved. So that's really what AlphaFold did. That's what all deep learning models do. And again, just some background. Neural networks, they're composed of hundreds of thousands of these artificial neurons. And you're going to see these architecture diagrams at varying levels of abstraction in this talk. Each neuron you can think of as performing a kind of linear regression. But the key difference is that these neurons that perform linear regression are stacked and interleaved with nonlinear activation functions, which essentially just means that you're not doing linear regression anymore. They can learn nonlinear patterns, and they've been shown to be extraordinarily successful in a wide variety of tasks. If you want a kind of mental model of what this is doing, you can start with the left-hand side, which is what is actually happening. You have these circles which represent numbers in your computer. These are all the parameters that are being adjusted. An input goes in X, some processing happens, which is basically additions and multiplications, and then the nonlinearity being applied, which is another very straightforward function. And at the very end, you get some output. If you really, really push the analogy, which I don't want to push very hard, you can imagine that there's sort of a stimulus, and it's as if it's going through some biological neurons, and you get some kind of response. But in the end, deep learning is really not particularly an accurate representation of anything happening in a human brain. But the human brain is a source of a lot of inspiration for deep learning. And I really want to put AlphaFold within the broader timeline of deep learning. Deep learning was not always the success that it was today. Back in 2011, it was really a very risky research field, if anything. 2012 was a time when things changed dramatically with the publication of AlexNet, image classification model, a deep neural network that looks at images, like pictures of cats, pictures of mushrooms, and just says exactly what they are. And this was the state of the art in 2012, and it blew away the competition, and it made people really realize that deep learning was a serious contender. In 2016, you might have heard of AlphaGo. Go is a board game that has many, many more possible moves than chess, so it was significantly harder to solve than Deep Blue beating Garry Kasparov in the 90s. That was solved by essentially a brute force search optimization problem. People handcrafted the rules for how to play chess, and the computer won by basically counting every move and seeing which is the best move. This was not feasible in Go. And in fact, AlphaGo solved the problem in a completely different way. It played Go against itself millions and millions of times, and it learned the rules. It learned the strategies essentially by self-play. And a neural network did this. It wasn't a human expert that wrote down what the best way was to play Go. It was really all by deep learning. And AlphaFold was really along that same line, the same ideas that carried AlexNet and AlphaGo to completion, and in fact, the same company that launched AlphaGo really applied this methodology to protein structure prediction. Demis Hassabis thought it was, a very, very good idea to a right problem for machine learning. And then, it kept going. Obviously, now you have nonsense images on the internet, and you have ChatGPT, which you might have heard of. And the main thing I want to get across is that the same deep learning methodology here underlies all of these applications. So when you think of AlphaFold, think of two very big strands in research coming together. One is the PDB and the accumulated endless tens of thousands of hours of work to populate that PDB. And the other is really deep learning. I had the pleasure of speaking to John Jumper about the development of AlphaFold, and you really understand that not only is he a computational physicist and a biophysicist, but he's really a deep learning first person. AlphaFold is first and foremost a deep learning break through. And to understand AlphaFold, you need to understand deep learning. To understand BioEmu, you need to understand AlphaFold, which is why I'm starting here. Yes.
I had a conceptual question maybe. So AlphaFold has been trained with all this amazing data that have taken, I don't know how many years of work, 100 years, maybe 1,000. So how do you think that this will progress starting from now, where people are using more and more predictions and maybe not depositing, real data at the end?
That is a fantastic question. I mean, in many ways, the data overhang is spent, right? So it took decades to create the PDB. The PDB has been milked to completion, and we're out. That's in. So from one perspective, I don't know what to do. If I knew what to do, then maybe I would have solved it. From another perspective, there are actually things that we can do with neural network predictions by treating them as data as well. And you'll see later that actually BioEmu was trained on predictions from AlphaFold. And this seems kind of silly. It seems like you're kind of recycling the same data over and over again. But there are ways that you can actually inject information into this process to make it a useful endeavor. So I'll go to that. A lot of machine learning now is thinking about how do I train on things that have come out of another neural network in a clever way. But it's definitely a much harder problem than it used to be. It's almost as if the PDB was a sponge that was wet, and then AlphaFold squeezed it, and then now we got all this great stuff out of the PDB, but it's kind of done now. We don't have extra data.
And do you think that people should continue doing, like, I'm looking at math, doing crystallography or because these are real data. Like this, sure, if we have crystallography of a molecule, we are sure that it's happening. So how do you think that we should?
There will always be a place for experimental information, but I think AlphaFold tells you, in a sense, where to look. So there is no, AlphaFold gives you confidence measures. Often these confidence measures are fairly decent. And so you can get some guidance into what AlphaFold knows really well, what's very similar to what's already in the PDB, and is very likely to be accurately predicted, and where AlphaFold is making a completely blind guess and it's nonsensical. And anybody who's working AlphaFold for a while has some kind of intuition for what it does well and what it doesn't do well. And all these experimental techniques are going to survive, but it's going to make more sense to work on the ones where deep learning doesn't work right now, and that's where we're going to get information.
Before you get too deep into it, I was going to ask, I don't even know for AlphaFold, but also for your platform, post-translational modifications. So, right, there could be phosphorylations, there could be silations, there can be so many, probably ones we haven't even thought of yet on lots of different amino acid residues on any one given protein. So when things are solving structures or thinking about dynamics, how is that taken into consideration? And are you able to perturb that to say, no, I want this residue to be phosphorylated in the model?
Yeah, the quick answer is they're not. So AlphaFold 2 doesn't give you any opportunity to control post-translational modifications. AlphaFold 3, I think, gives you some limited ability to do that. I don't know how accurate it is. BioEmu ignores this problem completely. Basically, the distribution of things crystallized in the PDB, that's kind of what AlphaFold 2 and BioEmu are trying to solve. And to the extent that PDMs are not involved in that process, that's kind of, they're blind to that. Yeah. Also, I don't know much about PTMs, but I know that the AlphaFold doesn't take them into account.
You can, in AlphaFold 3, you can select certain post-translational modifications. Yeah. It's not all of them, and it's a little finicky, but in AlphaFold 3, you can put post-translational modifications on a specific residue.
Matt says he doesn't believe it.
He doesn't believe anything. But my assumption is not an assumption, there's actual evidence, right, that post-translational modifications impact structure. So that's what I was kind of getting at is
it's a gap.
The models get worse the more complicated they are. AlphaFold 2 is more complicated. The thing you're trying to get a structure of is AlphaFold 2 is really good at monomers. AlphaFold 3 is also very good at monomers. They get worse when you have protein-protein interactions. They get worse when you have protein-ligand binding. They get worse when you have post-translational modifications. Sometimes they mix up the hollow and apo states. The more kind of layers there are to the problem, the worse it does. And there's some insight on why AlphaFold 2 actually does much better for monomers. I'll get to that later. All right, so they won a Nobel Prize. AlphaFold, how does it work? So multiple sequence alignment is the first step in running AlphaFold. So, yeah, who here knows what a multiple sequence alignment is? Okay, most of you. So, right, so you have, when you run AlphaFold, what it does is it does a genetic database search. It creates this MSA, and it's really just a way of associating amino acid sequences of related proteins of different species. And the key insight that's useful for AlphaFold is that MSAs provide information about structure, roughly speaking, because maintaining consistent structure is critical to how the protein functions, and amino acids that are critical to that structure get conserved across evolution between different species. Those that are not vary across evolution. So by looking at how a protein that does the same thing in many different species, which amino acids vary, which ones don't vary, you get some idea of what amino acids are crucial to the structure and which ones are not. And that gives you some understanding of what the protein might fold into. AlphaFold exploits this because the input to the network is not the raw sequence. It is this entire MSA. And the MSAs are processed by something called the evoformer, which is a neural network module. Evoformer is a name they made up. But it's worth thinking about this a bit because John Jumper, when he explains AlphaFold, the evoformer is the thing that made it work. Everything they had tried before introducing the evoformer didn't work very well. The moment they put in the evoformer, basically the problem was solved, and then they were tweaking it. So that's really the key. And to understand the evoformer, I need the first thing I'll talk about is that it's really just a variant of the transformer. So you might have heard of ChatGPT. The T in transformer stands for, sorry, the T in ChatGPT stands for transformer. It's a generative pre-trained transformer. I won't go into any detail here, except to say that this idea of transformers, it came about in 2017 in a paper called Attention is All You Need, which has been cited, I don't know, 150,000 times by now. And at a very high level, which I won't dive into, you can think of it as allowing a neural network to pay attention to certain parts of the input and pay less attention to other parts. When you're doing language generation, you can imagine that you have a sentence. And when a sentence is being generated, the different words, some words in the sentence are relevant to that word and some are less relevant. So like in this sentence, the animal didn't cross the street because it was too tired. The word it refers to the animal. So you can see that in the attention mechanism in a transformer, the animal lights up when you're trying to create the word it. Now, I'm very aware that this is not at all a rigorous definition, but that's as far as I can get into here. And just take away the intuition that you pay attention to certain parts, you don't pay attention to other parts. And when I talk about all this, at the very base of it, think back to those neural networks in the first couple slides. In the end, it's just numbers that are being processed by linear regression, and then every now and then there's a nonlinear function. These are all just functions, but we visualize them in different ways. So instead of attending to words, the neural network in the evoformer, it attends to amino acids in the MSA, which makes a lot of sense. That's the unit that's going in. And they're really the most important ingredient to AlphaFold success. And the main thing that, I'm not going to be able to explain the whole evoformer architecture, but the main thing I'm going to say is that it is a specially designed variant of the transformer that takes into account intuition that John Jumper and his team had about what parts of the MSA would be useful for predicting structure and how they would relate to each other. And there are three things that happen in the evoformer that don't happen in a normal transformer. Three ways of paying attention to the MSA information. The first is something called triangle self-attention. This diagram is in the AlphaFold 2 paper, and it's actually really hard to understand on its own. But the main point is that in standard attention, you have things attending to other things. So if you have a sentence of words, you have words attending to other words. In triangle self-attention, you're moving from pairwise communication. So pairwise communication would be, part A of the word talks to part B of the word. That's where the information is flowing, to group dynamics. And in group dynamics, basically, your attention is now asking the question, A and B both talk to C. So what does that mean about A and C? So like you have a triangle. So A, B, C. And think about this. In a protein structure, if you have 3 amino acids, you have amino acid A, B, and C. Not literally A, B, and C, but you have A, B, and C. If A and B are related in some way, and B and C are related in some way, that actually implies something about A and C because of geometry. It's the triangle inequality from mathematics, but that's really the intuition that John Jumper and co had when designing triangle attention. So that's a kind of specialization that they put into the evoformer that doesn't exist in a normal transformer. They also have row-wise attention. That's a lot easier to understand. That's just attention along a row of the MSA. So that captures the within sequence context of a amino acid. What is next to me in my sequence? And then there's column-wise attention, which is also easy to understand. That goes down the columns of the MSA. That captures the across sequence context. So how do I, as an amino acid, relate to related proteins in my position in related species? Again, I'm not going to go into the mathematics of how all this is defined, but the main thing to get across is that the breakthrough of AlphaFold happened because they took a standard neural network architecture. They thought carefully about where information is stored in MSAs and how it should relate to each other. They didn't prescribe a concrete rule, but they prescribed how information should flow through the MSA. And that's how they came up with the Evo formula. At the end of the Evo formula, there's something called a structure module, and its job is to take the process embeddings, so the kind of neural representation from the Evo formula and translate it into 3D geometry. Everything that's happening in Evo formula is happening in sequence space. Amino acids just attending to each other. There's no geometry yet. But at the very end, of course, you need a 3D structure prediction. So that's what the structure module does. I won't go into detail, but basically each amino acid in a protein is represented by a backbone frame, which is a position in 3D space, X, Y, Z, and orientation of the frame will pitch yaw. So if you have, a short chain of the protein, it kind of gets turned into these 3 little frames that the neural network is manipulating. I won't get into the details of the structure module because it turns out that the details are not very important. Between AlphaFold 2 and AlphaFold 3, the structure module was overhauled and simplified dramatically, and the performance actually improved. So really the heart and the key of it is the Evo formula. Everything after the Evo formula is just plumbing. So that was AlphaFold 2. I stress so much about AlphaFold 2 because much of this, basically all of this architecture is reused in BioEmu. But there's something that AlphaFold 2 doesn't do, which is predict more than a single structure or try to get at the equilibrium distribution. So as Nate has already said, knowing the folded state of a protein is not everything. And that's because protein function is often mediated by transitions between multiple conformations. So here's a little diagram from the paper of the example of the protein actin. And I don't really know much about actin, but the main point here is that the conformational changes that it undergoes when it binds to different ligands, they're what drive the function. They're what makes it tick. If you don't know these conformational changes, then you can't really understand what actin is doing and how it works. And to a large extent, there are ways to hack around this, but to a large extent, the original AlphaFold 2 and even AlphaFold 3 don't attempt to address this. They give you what they think is the most likely structure to appear in the PDB. And that's their approximation of the folded state. But this doesn't cover proteins that do interesting things when they have massive conformational changes. It doesn't cover things like intrinsically disordered proteins or intrinsically disordered regions of proteins or like domains that are rigid, but in between them you have a big weird linker, or local unfolding transitions. Like a lot of important things happen when a part of a protein that's normally structured unfolds in certain situations. AlphaFold doesn't try to model these things, and to the extent that it does so sort of accidentally, because sometimes in the PDB, the same protein is kind of deposited with slightly different structures because of different ligands attached to it, or because of just variability in the way that the experiment was conducted. And really, this is the gap that BioEmu is trying to fill. And I'm going to throw in some mathematics, but don't worry if this cannot be followed. I'll also explain it in English. But mathematically, you can write this. You can write down something called the Boltzmann distribution, which is a probability distribution, which tells you the probability of each conformation. So in this funny equation, X is the 3D conformation of the protein. P of X is the probability density. So if you know P of X, you know what shapes the protein takes and how likely it is. U of X here is the potential energy of a conformation. So where would you get that? Well, if you had a good classical model of the energy of a protein, the energy if you stretch the bonds, the energy if you kind of do that other thing that you can do with bonds and change the angles or like have a torsion, then you would know U. In practice, actually, it's quite hard to know what U is very well, but let's say that most of the time we have a good idea of what U is. K is Boltzmann's constant. T is a temperature. If you have a high temperature, the protein wiggles around like crazy. If you have a low temperature, it stays roughly in its folded state. Z is a normalizing constant, which you don't need to worry about. The only reason I show this equation is to say that we, through physics, we have an ability to kind of describe what the distribution of proteins are. We know that there's a distribution. We know that it depends on temperature. We know that it goes through lots of different, there's different modes, there's different valleys and things like that. If we could only sample from the Boltzmann distribution, we could describe every possible state of a protein and its relative probability, which would be really, really nice. Turns out it's extremely difficult to sample from the Boltzmann distribution, except for tiny, tiny proteins. Before I go here, I'll say a bit about molecular dynamics. So BioEmu is really, as was suggested in the earlier question, a kind of poor man's molecular dynamics. If you had, like maybe D. Shaw research, like a massive supercomputer that only does molecular dynamics, and you run it for many, many days on one protein system, then you can approximately sample from the Boltzmann distribution and answer any question you like about its distribution to within certain kinds of accuracy. This really works by simulating what happens in the body. So in the human body, you have a protein, it's wiggling and jiggling, like Feynman said, and it's wiggling and jiggling because of thermal fluctuations. So the other atoms around it have some temperature, they're bumping into the protein moves about, and that's what drives conformational flexibility. Okay, apart from ligand binding, which is another thing. That's what drives conformational flexibility in the first place. Certain states of the protein are more stable than others, and it will randomly walk into that state. And then it will stay in that state for a long time if it's stable. But then every now and then it will be jiggled in exactly the right way and then randomly walk into another state. This process is extremely inefficient for exploring the different states because that's not what the molecule is trying to do. The molecule is just being hit randomly. Molecular dynamics models this random hitting in thermal motion. So you basically simulate at every single femtosecond, which is 1/10 to the negative 12th of a second, every single femtosecond, how does each molecule jiggle a little bit? And you do this again and again and again and again, and you slowly, by a very, very, very long random walk procedure, enter one state, and then you get trapped there for ages, and then you enter another state, and you get trapped there for ages, and you enter a third state. And you hope that you've run this simulation long enough that you see every state, and not only do you see every state, you see how energetically favorable each state is. The way that I've described it has tried to emphasize how laborious this is, and how it is, it's great if you have 4 or 5 systems that you care about and you're willing to pay for them, but it is an inherently unscalable operation to perform. You cannot do this for, a kind of proteome-wide study to do like 20,000 proteins. What is the relative stability of each protein? There's this, basically, even with all the advances in GPU compute that we have, it would be utterly impossible. So there's a space for cutting through and turning this into a supervised learning problem. The analogy here is that in AlphaFold 2, we could get structures by X-ray crystallography or something like that. It's really laborious. We've done it a lot of times, but let's not have to do it every single time. Let's train a model to do it. Same thing with MD. We've run a lot of MD simulations. We've burned a lot of compute to do that. Let's not have to do that every single time. Let's train a model to predict the outcome of an MD simulation. That was the motivation for BioEmu. And here's really the goal in a nutshell. You get a protein sequence, you push it into this EMU picture, and then you get an equilibrium distribution, which is, you get to sample from it and you get to see the different conformations. And, there's some applications that we hope it would help. You can understand molecular mechanisms, hopefully. Hopefully you would help with structure-based drug design. Drugs, they're all about getting the protein to move a little bit, some part of it. And predict properties like, delta G, changes in free energies. Let me say these are aspirational. I don't want to claim that we've solved all of these. It would be pretty shocking if we did to perfect precision. So the first challenge here is training data. Data for equilibrium distributions is hard to come by. Molecular dynamics simulation data is very messy. It's siloed. It's not run like the PDB where you have 10,000, 100,000 structures, they're all deposited in the same way. Sometimes people run very short simulations for lots of proteins. Sometimes people spend $1 million in one protein. A lot of the data we have from molecular dynamics simulations is actually just of one protein, which is the SARS-CoV-2 spike protein. So it's a very inhomogeneous data set. That's a first challenge. It's hard to get by, and it's not all done the same. By the way, another big challenge, which is also why John Jumper doesn't like MD, is your molecular dynamics simulation is only as good as the force field that you use to parameterize the simulation. So when you're running a molecular dynamics simulation, you have to be able to say if I jiggle the atom in one way, what's going to happen next? And the only way to know that is to know some of the physics. And the only way to know the physics is to kind of run these simulations and match them with observed behavior of these proteins, and then tweak the physics a little bit until you get the right kind of parameters for like, okay, here is how springy a hydrogen-hydrogen bond is, or carbon-hydrogen bond is, things like that. This is a very long process of, typically it's called graduate student descent. And it's not perfect. These force fields are, only as good as what you put into them. There's no PDB for distributional data as much as we wish there was. So we had a kind of three-pronged approach that you could think of it as grabbing every source of data we could and kind of throwing it into a soup and hoping that deficiencies in one source would help out with deficiencies in another source, and then testing things at the end. So don't see a grand plan here, even though the paper makes it look like a grand plan. Don't tell Frank. Anyway, so the three-stage approach for BioEmu training data. The first stage is pre-training. I'll cover this later, but it's really training on outputs from AlphaFold, which is counterintuitive, but I'll get to that. The second stage is fine-tuning number one, which is basically we collected about 200 milliseconds worth of molecular dynamics simulation. Some of this comes from the internet. It's just free. Just open, you can download it. Some of it was done at Microsoft. We spent money running, on idle GPUs, lots and lots and lots of molecular dynamics simulations. We had real experts in molecular dynamics who ran thousands of simulations, set up the initial conditions, tuned the parameters of the force fields. They did things that, it was an enormous amount of the work was running many, many molecular dynamics simulations. And the third step was fine-tuning on experimental protein stabilities. This is the most interesting one in some ways. So we had a collaborator at, I can't remember which university, but it's Gabe Rocklin's lab. Gabe Rocklin developed an experimental assay that allowed him to approximately measure the protein folding stability, like delta G, of about 500,000 proteins, which included wild types and many, many of their mutants. So this is, indirectly, this gives you information about equilibrium distributions. It doesn't tell you what the structure is. It's not like he got 500,000 crystal structures, but it tells you, how likely are you to find this protein in a folded versus an unfolded state, which is relevant. Yeah.
Sorry, just a quick question. You say a lot of GPUs. Roughly how many GPUs are we talking about for the fine-tuning step, and for how long? Like just
for training or for data generation?
For the tuning part, not for not the data generation.
Yeah,
just tuning the
model.
model.
Think of dozens of A100s.
Around dozens, and how long roughly? Like, are we talking days?
Think of like days to weeks. Yeah. Yeah.
Thank you.
I think it's actually in the paper, but I can't remember. Like we ran many, many training runs. The final training run is a small fraction of the compute because most of the time things just didn't work. Yeah.
So is the data that was generated for these fine tunings available to researchers? Since you've spent
I think a lot of it is, yeah, it's been recently open source. And actually, if you look at the science paper at the very end of it, there's a link to how you can download that data. Yeah. Great. So yeah, so that part I won't get into. We developed a new method to handle that called property prediction fine-tuning. And the question is really, if I have a delta G value, which is just one number, how can I give that to the neural network in a way that allows it to understand the structure a little bit more, which is a bit of a tricky inverse problem. I'll focus on AlphaFold predictions in stage one. So stage one is what we call AFDB pre-training. AFDB stands for AlphaFold database. Basically, you start by downloading 200 million protein structures predictions from the AlphaFold database. So if you go online and search AlphaFold database, you can download all these predictions. Basically, they took 200 million sequences from UniProt. They just ran, by they I mean Google DeepMind. They ran AlphaFold 200 million times, they uploaded everything online, and we downloaded everything. That is not in itself very useful, because if you train a model on that data, you will recreate AlphaFold, which nobody needs another AlphaFold. So what we did instead was cluster these proteins by sequence similarity. So a lot of these have some amount of sequence similarity to each other, and we just put them all in a bag. So that's the bag. And what we did was we had a procedure for grouping diverse structures with similar sequences together. So often AlphaFold, if you make small changes to the sequence, it actually produces fairly significant changes in the structure. And you might wonder why, AlphaFold wasn't trained to do that. But again, this comes from the kind of natural uncontrolled variability in the PDB. Even for very similar structures in the PDB, sometimes there is genuinely a large conformational change. Sometimes it's actually ligand induced and AlphaFold doesn't realize. Sometimes it's just because of the experimental determination method, the way that the experimentalists resolve the X-ray diffusion, like electron map into things. There's variability in that. And that's actually like kind of a hidden information in AlphaFold that it kind of throws away when you run it normally. When you run it normally, it gives you one structure, but actually you didn't realize that if you tweak it a tiny bit, it will actually give you a completely different structure. And we want to make that explicit. So we group things by sequence, but divided it out into different structure kind of categories for each sequence. And the consequence of training on this data is that we force BioEmu to learn conformational variability. So basically say, when I give you one sequence, I'm going to give you 3 kinds of structure, and you need to predict all 3. Or I'm going to give you 5 kinds of structure, and you need to predict all 5. Don't just predict one. That's how AlphaFold is trained. I want you to predict all. And that's really why we did this. So that's all the training data. That was a whirlwind tour of that. But now I'm going to spend a little bit of time talking about the diffusion model. So I've mentioned already, AlphaFold, one structure for one sequence. Diffusion models are different. Diffusion models are a way of using neural networks. You can think of them as a kind of neural network. But they output distributions over data. So roughly speaking, the input to the diffusion model is random noise. So like on the right of this image, you have random noise. It's like, static from a normal distribution. And the diffusion model is trained to slowly and iteratively, step by step, turn that noise into an image of a data image that resembles some of the data in your training set. And there's so much that I could say about diffusion models, but in the end, the only important part for this talk is that the variability in the noise translates into variability in the output. So if you gave it a different set of random noise, it would give you a different dog, I guess. But for us, if you gave us a different input of random noise, we get a different structure out. And the model has been trained to say that I'm not going to always give you the same structure. I'm going to give you, 3 structures, and I'm going to give you the first structure twice as likely as the second and third structure because I saw the first structure twice as often, that kind of thing. So this makes diffusion models an ideal candidate for a Boltzmann distribution emulator, which is what BioEmu is trying to be. And here's like a fun GIF that I didn't make, that I got from the internet, of diffusion model generating protein structure. So the start is the kind of random noise structure. No protein looks like this kind of messed up nonsense, but it slowly denoises until in the end it's actually a designed protein that maybe exists. So that's, I won't go any further into that, but now I'm going to just very quickly talk about some things that BioEmu can do. One of the things that we benchmarked it on was domain motions. So this protein, adenylate kinase, I don't actually know what it does, but it has 2 different conformations and they're related by a large-scale domain motion. What we do is basically we sample from BioEmu 1,000 times or so, and then we plot on 2 axes the RMSD to each of the conformations of the PDB. And we noticed that some of the samples are very close to 4 AKE, 1 AKE. And this is 1 AKE, which is one of the PDB deposited structures, and a lot of the samples are really close to 4 AKE, and some are close to neither. So the main thing we wanted to see here was that if you sample for BioEmu enough times, would you be able to capture both of these conformations or not? If you ran AlphaFold 2, you'll get 1. There are some tweaks that you can do to like edit AlphaFold 2 in a way that you can maybe get more of these, but we wanted to systematically benchmark over many, many, many kinds of proteins. If you run BioEmu, sample from it about 1,000 times, you capture all of the interesting conformations that we know about already. By the way, these are all test proteins, so these are all proteins that were not in the training set. They're also not sequence similar to what was in the training set. We have a sequence similarity cutoff of, I think, I can't remember, look at the paper, but yeah, there's some sequence similarity cutoff to make sure that we're not basically training on the test site. Yes?
What does 1 AKE and 4 AKE mean?
Yeah, so these are 2 structures deposited in the PDB for the same protein, adenylate kinase, and in one of these cases it's open and one of these it's in the closed state. So we know that both of them are experimentally, observed structures, and we want to see if BioEmu can approximate each of them. Yeah.
Out of curiosity, in AlphaFold, if I put in an amino acid sequence and Nate puts in the exact same sequence on a different day on a different computer, will it always give back the exact same structure?
So in AlphaFold 3, no, for sure, because of some technical reasons to do with the fact that it also uses a diffusion model, but it's not actually using it to predict equilibrium. For AlphaFold 2, that one I'm not sure, honestly. I think actually there's random seeds that you can control. And when you run AlphaFold 2, you actually get back 5 predictions or something, and it gives you like the top most confident prediction or something like that. And I think that has to do with there are various places in the forward pipeline where you need a random number, and it's not like a huge part of it, but it can affect the predictions a little bit. So I would say no for both cases. You get subtly different predictions. For AlphaFold 3, you probably get bigger differences than AlphaFold 2 just because of the way they constructed it. Yeah.
But any of these describe what is that, how stable or how long the protein will be in A state versus B state? That was measured in this?
Not in this experiment, but in 2 slides. I'll talk about that. Yeah. Great. So that's one kind of motion we care about. Another was local unfolding. So RASP21 is an oncogene, I'm told. So it has a helix that can unfold. And Frank really likes this protein because there's PhD on it. And basically what we're trying to measure on the x-axis here is the fraction of native contacts. So native contacts are basically in the folded state, which amino acids are touching each other. Those we call the native contacts. If all these native contacts are contacting, then the fraction of native contacts is 1. And if very few of them are contacting, then the fraction of native contacts is 0. And what BioEmu predicts in this case is that there are actually this like gray thing is a distribution. It's a bimodal distribution. Half the time, you're folded. You have all the native contacts, and half the time it's just this helix is just completely, randomly arranged, which is what we expect. We then do a comparison with molecular dynamics simulations. So on the left here, I'm showing structures. They're labeled 1, 2, and 3 for 3 different kinds of protein. These are all small proteins, homeodomain protein G, BBA. And on the right, I'm showing TICA plots, time lag independent component analysis. What really matters is that the right is a distribution, is a visualization of the distribution. You can think of it like PCA. And it's a heat map. So areas that are more blue, that's where more of the conformations lie. So if you look at MD, for example, let's look at this one. So MD says that this is what the distribution of structures looks like. And there are 3 regions that are more dark blue. So 3 regions have more density, have lower free energy. And then if you look at region number 1, that's actually just the folded structure of this protein. If you look at region number 2, it is a somewhat partially unfolded structure, and region number 3 is just pretty unfolded. And what we get from BioEmu, which is this column called DesRes fine-tune, don't worry about that. What we get from BioEmu is basically instead of running a molecular dynamics simulation to generate this heat map, we just sample from BioEmu many, many times. And the important thing about BioEmu is that when you click sample, you can do that in parallel. So you can sample 1,000 samples in parallel. You don't have to sample one sample and then do a simulation sequentially based on the first sample to get a second sample, which is what molecular dynamics has to do. So MD, to generate this, I think this was done by DesRes maybe like 10 years ago, actually, but still right now it would take several thousand GPU hours, I think, to generate all these simulations. For BioEmu to generate this approximation, which, it's not perfect, but it gets some of the major features correct. For BioEmu to generate this approximation takes about, like, I don't know, a minute or 2 for each of these proteins. And then finally, yeah.
So if it takes a minute or 2, if you did a image sample, would you get better precision?
Yeah.
You denoise a little bit.
So yeah, that's a great question. So there's a kind of a bias and a variance thing going on. There's an inherent bias because BioEmu is not perfect. So if you get infinitely many samples from BioEmu, you get the precisely wrong answer in a way. If you have very few samples from BioEmu, you get a roughly wrong answer. And the question is, how wrong is that answer? And in this case, we sample enough times to see, we don't think that sample variability is going to control much more of what's coming out because every sample is IID from BioEmu. But we can see already in many ways that it's kind of wrong if we take MD as our reference point. But if we sample many, many more, we'll get a much smoother version of this. But I don't think the overall shape would change. Yeah.
Yeah, I would have been curious to see if you run like an hour with MD sampling.
Yeah. So MD sampling and BioEmu sampling, the sampling problem in BioEmu is basically benign compared to MD sampling because you can sample everything IID. So the sampling problem in BioEmu is like you're sampling many times from a normal distribution. If you sample like 1,000 times, you see the bell curve. But for MD, like because it's a Markov chain, you could sample for 1,000,000 years and not be sure whether you get the right shape. BioEmu, like we can be very sure that this is basically the shape that BioEmu is going to predict, but the shape might not be perfect. Yeah. Great. So free energy predictions. So here we take lots of these short proteins. Remember I told you about that data set from Gabe Rockland's group where he has 500,000 proteins, pretty small peptides really that he knows the folding free energies of from an experimental assay. So we hold out some of these proteins and we ask BioEmu to sample them. We look at the structures. We count how many of these are folded, how many of these are unfolded. And then, from the relative probabilities of folding and unfolding, you can compute a folding free energy, delta G. So delta G is really just a ratio of probabilities with a logarithm and some Boltzmann's constant stuff. All of it is just thermodynamics in the end. And you can get thermodynamics from structures. If you know all the structures, if you have all the samples from structures, thermodynamics is just, some processing of those structures. So that's what we're doing here. And the main thing to get is, this is not at all perfect on a straight line. But it's much better than a random chance ranking of the folding free energies and also the mutant stability changes. Yeah, if you're interested in how this relates to similarity to the training set, we've also plotted that here. The dark circles are more similar to things in the train set, and the light circles are very dissimilar to anything that was in the train set. And the main conclusion that we're trying to get here is that BioEmu samples reflect to some degree, at least a relative ordering of the stability of protein mutants. So if you have a protein, you're like, I'm going to mutate this one amino acid. Is it going to become more or less stable? BioEmu can give you some kind of prediction on that. Don't trust it 100%, but it's going to be better than a chance guess. So the final option is please try BioEmu for free today if you are interested in any of this. That's what I'm selling you, I guess. So it runs in the browser. You don't need to install anything. Just input a sequence and download the sample structures. It'll be done in 10 minutes. Yeah. Go to this link, I guess, or you can just Google BioEmu collab fold. If you ever use AlphaFold via collab fold before, this runs in exactly the same way.
So quick question. I noticed number of samples you input 10. Is that kind of a standard suggested number or should we change that?
Yeah, it's a default. I think what you want really depends on. So I think just imagine it's a bit of a kind of a cheap, weird MD simulation, and you're picking random points along the MD trajectory.
But when it says number of samples, it's going to give you 10 different.
Going to give you 10, yeah. Going to give you 10 structures that you can download.
3, it would only give you.
It'll give you 3, yeah. So you really want quite a lot to get an idea, because if you randomly sample from a trajectory and you only took 10 samples, maybe you missed one important mode that was just not very likely. So maybe I would ask for 1,000 usually to get a sense. I don't know how long that would take though. It's all running on Google's compute. But if you want to, you can run it locally too. It's just more pain.
10,000 PDBs, or is it some distribution of energy?
PDBs. Yeah, PDB files.
Now you have 10,000 PDB files to deal with.
Not hard. You just so if you're familiar with molecular dynamics, like data analysis, exactly the same thing.
Okay,
yeah.
So then you do clustering on that.
You do whatever you want with it, and basically, so the
idea that you're.
Yeah, so the hope is that this will be a stand-in for MD. So imagine you had a structure and you're like, oh, I wish I could run MD on this for 10,000 hours, but I don't have the compute. You do this instead, and you get a poor man's version in 1 minute. Yeah.
A really naive question. If I have a PDB file, what do I need to open that file?
I would recommend
a piece of software or something.
Yeah, I would recommend another.
You have a map there.
You don't need a Mac or anything. I would recommend using Molstar. Molstar, MOLSTAR. It runs in the browser, so you can use it on Windows, and it makes beautiful visualizations. Purists will go for PyMol. PyMol is hard to use.
Yeah, I don't want to.
Yeah, so if we are interested in drug development, right, and traditionally we start from crystal structure, maybe compound docking, to now AlphaFold structure, gameplay if that's close enough, compound docking. How do you envision your can generate different states, right? And then how do you see these can boost up the new drug discovery that was unthinkable before?
Yeah, again, I'll put a lot of caveats on this because it's not a perfect model and we really wish it was better and it doesn't handle, complexes, which is a huge limitation. But one hope that we had in running this, and then you can see benchmarks on the paper and how accurately it does this, is that it will help us find cryptic pockets. So if you have a protein you're trying to target with a drug and you don't find any surface that a drug would attach to, you can hope that there's a cryptic pocket. You run it through BioEmu, it gives you suggestions for, if BioEmu gives you 10,000 of the same structure, then maybe you're out of luck. But maybe, 100 of these structures actually have a little thing opening and you're like, wow, maybe that's a target where a drug could bind. So that's really what we were hoping. Yeah. But you can look in the paper for a detailed explanation of how accurate we are at guessing cryptic pockets. Yep.
I was thinking about protein design.
Yeah.
Experimentally, for example, in the lab, sometimes we want to use something to a protein,
Yep.
and we just try an error. We put a tag in the terminal, C-terminal, see if it behaves well.
Yeah.
If this tool could actually predict if a tag of a certain size in specific sites of the protein.
Are you trying to design a binder for another protein?
Yeah, it could affect the interaction with other protein or
Uh-huh.
the actual folding of the protein and may affect the function if this could predict. So you would never put a tag in there.
Yeah,
Put it on the other side.
So it depends a bit on the question you're trying to solve. If you're trying to design a binder, BioEmu in its current state is not going to help you because it can't do complexes. But I'm told that the second version will address that. Right now, if I wanted to design a protein binder, I would look into something called RF diffusion or RF diffusion is a good one to start because it's.
This question is more if you just fuse on the domain, will it ruin the fold?
Oh, I see. Oh, so it's not a binder. It's like part of you're just editing the chain.
Additional
Yeah.
tag. Flag tag.
Yeah, BioEmu is a good thing to try. Like again, don't trust it as gospel, but it will. The good thing about BioEmu is that number one, it'll give you some prediction, which will kind of guide you a little bit, and number two, if it gives you a prediction, it will give you a mechanism for that prediction. Like it will say it will show like, oh, this reduced the folding free energy. You can inspect. That's because in 10% of the samples now, you see that this amino acid is now being repelled by this other thing, and that gives you structural insight that you can actually then carry forward. So yeah, I would totally. If what you're trying to see is like, if I edit this sequence, will I destabilize the fold? This is a perfect use case for BioEmu. Yeah, and you can run that experiment in 10 minutes.
Wait, how much would it? Can we run it on like a Mac? Or is there enough power?
I think you need an A100 probably to run it locally. I think you might run out of memory on a Mac 600, but I'm honestly not sure. Yeah, I've never tried to run it locally. I think if you're trying to run it locally, then you're not really processing a lot of proteins, a lot of big proteins. So actually CollabFold is a better option. But if you really want to process a lot of proteins fast, then you really need something like an A100 to run this. Yeah.
So nice.
You know,
Questions for Andrew?
Looks like we have another good tool.